Welcome to the CRISPR-Cas video series of the project Genetic Engineering and the Environment. My name is Katharina Kawal. I have a doctoral degree in molecular biology, and I will explain the technology, the possibilities, and the risks of CRISPR-Cas to you in different videos. These videos describe the content in an easy to understand way and are aimed at everyone who wants to be informed in an objective way. In this video, I start with the first part describing the technology. Here's an overview of what the new genetic engineering techniques or genome editing actually are. These are the site-directed nucleases such as CRISPR-Cas, the TALENs, or the zinc fingers. These technologies are able to cut at specific regions of DNA, therefore they are also called gene scissors. Oligonucleotide-directed mutagenesis, which is abbreviated as ODM, is also a new genetic engineering method. The focus of this video is on CRISPR-Cas as it is the most commonly used genome editing technique at the moment. The other techniques follow similar principles and are not further discussed here. I will also focus on the use of CRISPR-Cas in plants, but the technique is also used in human and animal cells and in microorganisms. Some biological basics are important to understand how CRISPR-Cas works. This picture shows a cell nucleus with DNA. The DNA is made up of sugar, phosphate groups, and DNA bases. The four bases adenine, thymine, guanine, and cytosine determine the DNA sequence of genes. The complete DNA sequence of an individual is called the genome. The double-stranded DNA is coiled up in the cell nucleus and is organized in chromosomes. Before I explain to you how CRISPR-Cas works, I would like to show you how the gene scissors get into the plant cells. CRISPR-Cas can be introduced in various conditions. One possibility is to bring in the DNA that carries the genetic information to form the gene scissors by the plant cells. This circular DNA is also called a plasmid. The gene scissors themselves are formed from this DNA. The gene scissors are an enzyme complex consisting of the cutting component Cas and a recognition component called guide RNA. The guide RNA guides the gene scissors to the place in the genome where it is supposed to cut and is designed to recognize a specific DNA sequence of the target organism. CRISPR-Cas can also be introduced as an already formed enzyme complex. To make things easier, I'll show the enzyme complex as gene scissors in the next slides. A cell with a nucleus is shown here. Either the DNA for forming CRISPR-Cas or the already formed gene scissors are introduced into the cell nucleus of the plant cells. The DNA of the gene scissors is introduced using the old genetic engineering techniques, primarily with the help of the soil bacterium Agrobacterium tumefaciens in a biological process. The DNA can also be introduced by the physical method of particle bombardment or by treatment with the chemical component PEG. Already formed gene scissors are introduced either by particle bombardment or by PEG. If the respective conditions of the gene scissors are in the cell, there are different ways in which CRISPR-Cas becomes active. The first option is to integrate the CRISPR-Cas DNA into the genome. At the moment, this is the most common way of introducing the gene scissors into plant cells. The CRISPR-Cas DNA is firstly integrated into the genome, here into the blue chromosome. The result is a transgenic organism that carries the genetic information for the formation of the gene scissors. The gene scissors are formed and cut the target sequence, here on the green chromosome. In the last step, the integrated CRISPR-Cas DNA is segregated through subsequent plant breeding so that only the alterations at the target sequence remain in the genome. However, it is also possible that unintended changes in the genome occur. I will explain more about unintended alterations in another video, about the risks of CRISPR-Cas. The second option is to introduce CRISPR-Cas directly as an enzyme complex. The enzyme complex is produced synthetically in the laboratory and then introduced into the cell nucleus as mentioned before. There it becomes directly active and leads to alterations at the target sequence. After a while, the gene scissors are degraded by the cell. Attempts are currently being made to use this option in many different plant species, since the genetic information for the formation of the gene scissors is not incorporated into the genome and no transgenic organism is created temporarily. Nevertheless, even with this variant, unintended changes in the genome can occur, which I will explain to you in the video about the risks of CRISPR-Cas. Now we come to a more detailed explanation of how CRISPR-Cas actually works. The enzyme complex is now in the cell nucleus, where the DNA of the cell to be changed is located. The guide RNA is a short piece of RNA that has been individually designed by the user to recognize a specific target sequence. A so-called pump sequence in the genome of the target organism serves as a stop signal for the enzyme complex. It is a specific DNA sequence and it lies adjacent to the target sequence on the genome. The gene scissors stop at the pump sequence and use the guide RNA to check whether the DNA sequence in front of it matches its sequence. If this is the case, CRISPR-Cas cuts at that part and mediates a DNA double-strand break. The double-strand break is a DNA damage and an alarm signal for the cell. This signal activates the cell's own repair mechanisms to close the DNA damage as quickly as possible. The main function of CRISPR-Cas is to generate a DNA double-strand break. Everything else is done by the cell itself. After the gene scissors have caused the DNA double-strand break, there are different possibilities of what can happen. Two different repair mechanisms can be activated. On the one hand, that is a repair mechanism called NHEJ. The activation of NHEJ leads to a random and undirected repair of the DNA damage. The NHEJ repair is known to be error-prone. Either the original state of the target sequence is restored or small alterations are generated at the cutting site. If the original state is restored, there is the possibility that CRISPR-Cas will bind again to the target sequence and cut again there. So in the end, the target sequence will most likely be changed, resulting from an erroneous repair, which is the intention of the user. This type of application is also summarized under the term SDN1 and describes small, undirected changes at the target sequence. In addition to SDN1, there are SDN2 and SDN3 applications in which the DNA damage is repaired by a repair mechanism called HDR. HDR works through homologous recombination, which is an exchange of DNA sequences between identical DNA regions. For this purpose, DNA templates are synthesized in the lab and introduced by the developers into the cells in addition to the CRISPR-Cas components. The DNA templates correspond to a large extent with the DNA sequence around the target sequence. As a result, the HDR repair is able to integrate the DNA sequence of the DNA template with the intended alteration at the target site, shown here in orange. The difference between SDN2 and SDN3 applications is the length of the DNA sequence introduced with the template. With SDN2, only a few base pairs are introduced. With SDN3, longer pieces, for example, complete genes are introduced. In contrast to SDN1 and SDN2 and SDN3 applications, the double-strand break generated by CRISPR-Cas is repaired in a directed way with the aid of the template introduced and the activity of the HDR system. If you look at the various SDN applications in plants in the more recent literature, you will notice that mainly SDN1 applications are used. SDN2 and SDN3 applications are only used to a small extent. The explanation for this is that the HDR repair works inefficiently in plants and the repair of DNA double-strand breaks by the NHEJ mechanism predominates. Using CRISPR-Cas, different genetic alterations at the target site can be mediated. Most commonly, SDN1 applications are used to knock out genes by integration of small errors into their target sequence, so that the gene no longer functions. The gene function can be altered. This means that the alterations introduced by CRISPR-Cas can have an impact on the functions of the corresponding protein. Genes or part of them can be deleted. In addition, the gene expression can be altered, and new genes can be integrated into the genome by SDN3 applications. In the next video, I go into more detail about these possibilities and how CRISPR-Cas can be used to mediate complex changes in the genome of plants. In further videos, I will also talk about the risks of CRISPR-Cas. Thank you for your attention. You can find more information at the website of the project Genetic Engineering and the Environment.